Hello. Good morning, everyone. First of all, I would like to thank the organizers for inviting me to participate in such a great meeting, and it has been very beautifully organized. So today I'm going to talk about evolution of translation system, and it's sort of a next logical thing in line of all those events that have been previously presented this morning and lined up. And I would like to acknowledge all the members of Center for the Origins of Life Bioinformatics group who contributed to this work over a number of years. So today I'm going to talk about the translation system and how it's evolved. And about an hour ago, I've realized that based on the prescriptions given by Lauren Williams, I should not be allowed to give this talk to you today, because I'm going to talk about RNA molecule, I'm going to talk about a model, and I'm going to talk about using a cell phone, a smart cell phone as an analogy in my investigations. Well, luckily I have a license to do this job issued by another authority. So when in about 1968, Carl Woese, who was just assistant professor at the University of Chicago, he wrote an encouragement letter to Francis Crick, and specifically he said, if we are ever to unravel the course of events leading to the evolution of the prokaryotic cells, this can be done by using the cell's internal fossil record. The obvious choice of molecules here lies in the components of the translational apparatus. So while I agree with some of the concerns raised by Lauren during this morning, with this license, I feel that I'm entitled to proceed further and complete my talk. What we have in general as people who search for quest of life, many biologists, they study life on Earth by using fossils. Fossils of different living organisms. And this is how actually the very first tree has been constructed. That was done by Ernst Haeckel. The problem, however, is that using geological fossils as a source of information for the origins of life, it's not sufficient, because all these biological fossils are dated at 3.2 billion of years ago. But we can use, by the same analogy, we will use the ribosome for the reasons I will mention a bit earlier, but they are kind of listed by Carl Woese. So we will use a ribosome as a molecular fossil, and we will try to extract as much as possible information stored within ribosome, and we will try to interpret this in terms of evolutionary processes, molecular evolutionary processes that we know. So the analogy is simple in the first approximation. So let me at least give this first. So the ribosome has evolved by accretion process and it records its information within its own growth shells. This is basically similar to what you see in okay, so this is how it's similar to how the trees have evolved by accreting their layers and recording its own information. So we will use the same principle of peeling off those layers of information stored within a ribosome, and we'll try to interpret it in terms of some molecular and evolutionary processes. Okay. So, but let's start with a smartphone. And the smartphone in this particular case is a human ribosome. So what we know that from the extant ribosomes, actually these are the 3 generations of ribosomes shown in this figure. So the first ribosome is from E. coli, the second ribosome is from S3 which is a simple eukaryote, and then the next one is Homo sapiens. So we do see a number of accretionary events happen at the surface of the ribosome. And we do see extension of those helices. And apparently the ribosome grows and it increases in size by almost factor of 2, especially for the large subunit, starting from bacteria to archaea to eukarya. If we take a closer look into a particular region of a ribosome, here I begin with a certain RNA called helix 25. We can see how that region increases in size and how it grows. And there are a couple of observations. First is that there is a continuous growth as we go, and I basically described this. But second is that the older part is always contained within a new part. So basically, we can see that the ribosome, this is one of the proofs that a ribosome contains an evolutionary history of its precursors, from at least in the extant biology. If we take a closer look into what is happening at those expansion segments, we can see even some additional details. And here specifically, we can see the underlying region of RNA, which we refer to as a trunk, and the new region of RNA, which we refer to as a branch. And there is a clear distinction between trunk and branch, which is identified by insertion fingerprint. So we can clearly observe some of the insertion fingerprints, and we can detect the regions within RNA at which the insertion happens. So, and it was a great discovery a number of years. And then we ask a question: if we can see those insertion fingerprints at the surface of the ribosome, then can we actually identify these insertion fingerprints deep within the ribosome body and try to trace down the evolutionary processes happening before LUCA that led to emergence of ribosomal molecules? And the answer was obviously yes, except that it was difficult to see at some point, but with aid of some carrier classes we managed to do that, and we were able to identify a number of about 60 of those fingerprints in a large subunit and 30 fingerprints in a small set. So with that, we were able to break the all ribosomal RNA into the fragments, which we refer to as ancestral expansion segments. And then the question was, how do we put those expansion segments in order? And I will make some comments on that about it later, but first let me show you how those expansion segments and fingerprints, how you can see them within the core of the ribosome, the peptide transfer center. So you can clearly identify those expansion segments, you can put the pieces, you can put them together, and you get basically the story of a PTC origin. And as I mentioned, we can do this exercise for the entire LSU and the SSU. We had 30 for the small subunit, 60 for the large subunit. In order to make our description simpler, we combined those expansion segments into groups, which we refer to as phases of ribosomal evolution. And here are the 6 phases of the LSU and the 6 phases of the SSU. And this is how the ribosome seems to have grown over time if we follow these principles of the accretion models that I recently described. So far I've been only talking about the ribosomal RNA. Obviously, the ribosome is a macromolecular complex that consists of ribosomal RNA and ribosomal proteins. And of course, we also looked at those, and we noticed some specific regularities. First of all, we realized that deep inside of ribosomal RNA, we have only unstructured peptides that penetrate deep inside those IDR regions, intrinsically disordered regions of proteins that do not have any apparent secondary structure. Then there is an intermediate layer within the ribosome that contains some elements of a secondary structure. Then finally we get to a region that we have some very simple protein folds, and finally we have ribosomes decorated with a very sophisticated folds containing a bunch of alpha helical elements. So that was again first mechanical analysis of how proteins of ribosome are organized, and basically, this figure summarizes what I've just described you. The point is that there is a progression, an increase in the complexity of protein folds as it has been recorded by the ribosomes. Putting all this together, we attempted to provide a combined model of a ribosomal evolution in which the pieces of ribosomes starting from very short RNA molecules and short peptides or deoxypeptides would come and cooperate and collaborate together, forming some initial basic three-dimensional structures that would be further interacting with each other, resulting in a formation of a proto-SSU, a small subunit, and a proto-LSU, proto-large subunit. At some point, those two subunits would be joined together mediated by proto-tRNA molecules. And at some point along the evolution, this system would start being engaged in the production of proteins through simple decoding process, and later on it will be powered by energy coming from initiation and elongation factors, and resulting in the formation of the ribosome. So that's essentially what we have. We started to look a little bit deeper into evolution of proteins at the ribosome. One of our striking observations was that many ribosomal proteins and proteins that are involved in a translation system, like initiation and elongation factors, they look like beta barrels. They have a very simple organization. There are many, and those are shown in orange in this figure. So the beta barrels are, a lot of them are part of initiation factors, and there are many of them in a large subunit. There are a few of them in a small subunit. So, and among those beta barrels, we kind of saw two different kinds of beta barrels. One of them is called SH3 domain, and the other one is called the OB domain. Those beta barrels, they have a similar topology. Each of them contains 5 beta strands, but topologically they're different. They're not related to each other. So here we ask the question, if both beta barrels they are so abundant within ribosome, are they related to each other or not? And so far, there has been no evidence that those 2 beta barrels are evolutionarily related. So Claudia and Peter have set up and performed a study in which we analyzed the sequences and structures of those beta barrels using very sophisticated computational methods. So first we collected all different created structural library of beta barrels, we put them together, and you can see that indeed, if you ignore their differences in their topology, their scaffolds are indeed quite similar. And the key point was to realize that if you account for a process which is known as a circular permutation, basically you cut something and you move it to a different place, kind of circularly rearrange a sequence of one protein compared to the other, you will get a very good information in a sequence space between OB and SH3 folds. And that was our first guess that how we can possibly show that those 2 barrels are evolutionarily related in the sequence space as well. And indeed, when direct search by all sophisticated methods that I'm not going to be covering today did not provide any good results, the search in a circularly permuted space apparently resulted in revealed us some good relationship between some of the beta barrels representing OB and SH3 domains. Based on that, we proposed a model by which, so just having 2 domains related by circular permutation, it's great. We do know at this point that those are evolutionarily related. But it's not satisfactory to explain that those 2 domains can basically move one to another by simple rearrangement of their sequence, because at the biological level there is no such a mechanism. Proteins do not cut their sequence. Something has happened to a gene level, and we proposed a model which we call constructive destruction. And this model implies, it's a 2-step model, which involves a gene duplication followed by rearrangement of certain strands and refolding of a new sequence, forming a new seed, forming a new folding landscape, and forming a new fold. And this model satisfactorily explains interconversion of OB folds and SH3 folds. We attempted to justify the principles that we found by creating artificial sequences, circularly permuting sequences, and proving that those can fold by doing alpha fold modeling. So with that, I will stop, and here are my conclusions. Ribosomes are molecular fossils, and we can get some history of what happened, of early events that has been recorded by ribosomes. We can just unravel this by looking into data. We can do it by analyzing the RNA molecules. We can do it by doing analysis of ribosomal proteins. With that, I'm happy to take questions. Thank you.
Anton, I love your approach to this, but just to advocate, doing Goulian and intrinsically disordered proteins, they are also more flexible in their function, and I'm wondering wondering whether there could also be a functional explanation for the fact that you have these layers going from intrinsically disordered to higher and higher orders, that it has to do with having more flexibility and function in the inner core and then more stability in the outer core. It's just an alternative hypothesis for you.
No, this is a great question, and in fact, for sake of time, I sort of, yes, okay. This is where the details matter. What I did not mention, of course, I presented this work as if we were reading everything from the extant ribosome. Of course, you have to take it with some grain of salt. That's what this is Laura Williams is calling you, right? So what is happening is that so remember I mentioned that initially we have some peptides and deoxypeptides, and those initial pores have been created by those non-coded proteins, peptides, let's put it in this way. So what we see right now, our interpretation, so those non-coded peptides form those pores within body of the ribosomes, and we interpret those as molecular casts. And those molecular casts have been now filled by those intrinsically disordered tails of proteins that go and there was a molecular replacement process, replacing those non-coded peptides by IDR regions of intrinsically disordered proteins. Did I answer your question?
Not really. What I'm wondering is if you're doing this experimentally, for example, you could try replacing some of the outer ones with intrinsically disordered, or some of the inner ones with secondary structure. That again, there may be some sort of functional aspect. But it's
okay. Talking
it's
about
a difficult question.
Talking about the functional aspect, in fact, some of those IDRs are extremely important because BL27 in bacteria comes very close to the PTC, and it participates in the engagement of some of water rearrangement close to the PTC, and essentially participates in the peptide transfer. Doing a replacement of the others, if you compare archaea and bacteria, they have slightly different layouts of those, so that already an indication of some of the processes that nature had taken in a slightly different approach. But within each archaeal and bacterial domains, all those proteins are conserved. However, if you're going to later on to a mitochondrial species, that's where you get a lot of interesting phenomena, and I guess that may be related to your question, because in the mitochondria, RNA is getting reduced, and many of those inner part of a structure being replaced by IDR regions of mitoribosomal proteins that get much longer within the ribosomal cavity because there was this replacement. So they're taking those additional functions, replacing some of the non-functional RNAs. So we'll talk about it.
Additional questions? So maybe I'll ask. So, I mean, you had in one of your diagrams, you attempted, in some sense, to map this structural evolution to more functional evolution, but you didn't really talk about that in detail.
Right.
So, so maybe I'm giving you a chance to talk about that in a little more detail, because that's, of course, an interesting question: is exactly how some type of selection pressure is driving this this structural evolution that you identified.
So, excellent. Thank you for this clarification. So, I guess you're referring to this diagram, and obviously we try to assign a certain function to every stage, assuming that, yes, so nature doesn't have a foresight, and at each stage the selection would be driven by a certain function. And so if early on the function of deoxypeptides would be just to provide a stability to some of the folding competent RNA molecules and prevent them from dehydration. So then at stage 2, which is a light blue, we talked about emergence of the proto-peptide transfer pore, and at this point we assign this function for already ability to make some peptides by this cavity, which is known to be as the holy grail of the ribosome, the peptide transfer center. It's further getting encapsulated by additional RNA, and it results in a phase 3. And this is where at phase 3 we bring a small subunit into the story, assuming that initially a small subunit was just a simple cofactor that was enabling to keep two tRNA molecules next to each other at the PTC, such that the peptide transfer reaction within the PTC by integrating the CCA tail would be much better. So it's a synchronization cofactor. Once both subunits have been in place, then basically we have a proto-ribosome as in a normal way, and then from that point on it evolves as a simple ratchet machine towards phase 4, and then at phase 5, which is an early phase where it's actually emergence of coding and bioenergetics take place, and then finally we get to mature ribosome. Yes.
Thank you. That's good. Yes.
Great talk. I'm Petrov here from Sofia University. Can you provide a time frame for each of these phases? And I have a second question. Do I understand it right that on the first phase, the presence of maybe first few phases, the presence of magnesium is not necessary because we don't have second subunit?
Okay, so the time frame, I guess, it may all happen very fast, relatively, assuming it was somewhere between 4.2 and 4 billions of years ago. I cannot provide an absolute time scale. I can only provide you some relative time scale. Contrary to answer your second question, it's contrary to what you just said. I have not mentioned at all about this, at least from the analysis of the extant ribosomes, especially from early structures that are available to us from 2000s that have been produced by X-ray crystallography, not by cryo-EM, in which we do have all the information about ions. So it tends to be that more ancient parts of a ribosome are enriched with magnesium ions, and the magnesium ions, in fact, plays a role in stabilizing many interactions between different backbones. There are some special magnesium ions, and we have a number of papers that I will be happy to share with you later about those special magnesium ions that are critically important for ribosomal assembly and function, because those ions bring together a number of remote fragments from a ribosome and held them together in a very, very specific way.
Okay, well, so thank you again for